All right, you guys. I'm trying to record this, so any questions that you guys have will probably be on the recording, just so you guys know. If it works out, I'll upload it to YouTube so you can take a look at it. Hopefully that's okay. If you guys are not wanting to be recorded, just let me know, or maybe ask me questions after the review session. Anybody have any objections to being recorded? All right, so ask away then. Okay, so I didn't have enough room. This is what we're going to start doing right now. We'll continue on with the intrinsic mechanisms and the extrinsic mechanisms after we cover this, okay? So we're going to start off with the anatomy. Does anybody have any major burning questions right now about concerning the anatomy of either the urinary system or the nephron? Okay, so we can go through that right now if you guys would like. The system is pretty straightforward. I mean, this is just basic anatomy, general anatomy. You'll need to know basically these four different structures: the kidney, ureters, the bladder, and the urethra. Pretty simple. The major structures in the kidney are going to be the cortex region, okay, the cortex, that's the outer part of the kidney. The medullary region, the medulla, okay, that's kind of those, we did this close to Halloween. Those are the candy corn looking structures here. The darker regions are the medulla or the medullary region. Then I need you to know the difference between the minor calyx and the major calyx. Okay, the minor calyces are right here. They're these smaller, I can probably blow this up a little bit. These are the smaller regions right here, the minor calyces. The major calyces is the larger cavity before you get to the renal pelvis area. The renal pelvis is the area right before the ureters. Okay? So, going back to this figure, this is actually labeling the minor calyx and the major calyx here. And then the renal pelvis is like this area right here, and then the ureters. Yes?
So, when you're distinguishing between the minor and the major,
yes.
I'm still kind of having a hard time visualizing where each one is. Do they have a different function?
You know, they're just mainly the cavities. I would say the minor calyx is right next to the renal papilla. Okay? The minor calyx is right next to the renal papilla. Remember, the papilla is that inner medullary region, that point of the candy corn, if you will. Okay? So I would consider this is the papilla. And then the cavity right next to that, where all that filtrate is going, that ultrafiltrate, that's the next cavity is the minor calyx. And all of these minor calyces, they basically, the urine goes from the minor calyces into the major calyx. Okay, that's the larger cavity that all of those minor calyces filter into.
So is there only one region?
Yeah, I would say that one region really is, all the minor calyces are filtering into the major calyx, and then that filters into the renal pelvis. Then the urine goes into the ureters, okay? So I would say, please be able to label this, and also note the renal artery and renal vein, okay? Renal artery and renal vein. Now remember, the renal artery is going to branch into arterioles. Also, it's going to branch into the afferent arteriole, the glomerular capillaries, the efferent arteriole, and then it's going to branch even more into capillaries that perfuse the nephron. Okay? And then it's going to come back in the form of venules, and then the one renal vein is going to carry blood out of the kidney. Okay? All right, so taking a look at this nephron, remember, this is not to scale. This is ridiculous, okay? But it does show you that the glomeruli, I mean, there's hundreds of thousands of nephron in each kidney. The glomeruli are all pretty much in the cortex region. So note that glomeruli, they're all going to be in the cortex region. And then there's only certain nephron that are going to have loops of Henle that extend deep into the intermedullary region. Does anybody remember what those nephron are called? Yes, very close. Juxtamedullary nephron. Perfect. So juxta means next to, medullary, the medullary region, juxtamedullary nephron. Okay, perfect. Now, those nephron that do not have loops of Henle that go deep into the intermedullary region, what are those called? We have two different types of nephron we talked about in class. What's that? I did hear it. Cortical. Yeah. So the other type of nephron is the cortical nephron. So please know the difference between juxtamedullary nephron and the cortical nephron. Which type of nephron then is going to establish that osmotic gradient in the intermedullary region? The juxtamedullary nephron. They're going to be responsible for establishing that very profound osmotic gradient. Now remember, without that osmotic gradient, you have no capability of concentrating urine without those juxtamedullary nephron. Okay, does that make sense to you guys? Yes?
What are the cortical nephrons?
The cortical nephrons are still filtering blood. They're just, and they are, forming ultrafiltrate and urine. Ultimately, they're just not participating in establishing that osmotic gradient. Okay, so now I have a question for you. What is the name of the blood supply that surrounds the cortical nephron? What is the name of the blood supply that surrounds the cortical nephron? I probably only said this once in class. So if you don't know, that's okay. Do you guys remember peritubular capillaries? Okay. Those are very different than the blood supply, and I did go into a lot of detail on this one. The blood supply that surrounds the juxtamedullary nephron. What's that called? The vasa recta. Okay? The vasa recta is very important in maintaining that osmotic gradient in the intermedullary region. So again, this anatomy is just general anatomy, but it does help you understand the function of the kidney. Okay, so make sure you know these different terms. Yes?
So the vasa recta is like a set of capillaries?
It is a set of capillaries, yep. And it is perfusing the nephron. Why would we need to perfuse the nephron? Why do we need the vasa recta? Yeah.
For the counter currents.
That's a special property of the vasa recta. Absolutely. It's organized in a way so that it doesn't wash away that very important osmotic gradient. So it's organized so it maintains the osmotic gradient. But why have it at all? Why have it at all? Yes?
For secretion or reabsorption?
Sure. So that's a good point. I wasn't thinking about that, but sure, you have to have the blood supply surrounding the nephron for secretion and reabsorption of different molecules. Yeah, that's a very good point. What else, though? What's important? What other structures are important in the nephron? Yeah, keeping the tissue alive, okay? There's epithelial cells that basically make up the nephron. It's lined with epithelial cells. And they have to be perfused with blood, oxygen, nutrients. Okay? These epithelial cells I love they're able to modify the luminal fluid. Okay? So that's why we call this the primary urine, because, or the ultrafiltrate, because it's not quite urine yet. There's a lot of modification by those epithelial cells, right? You can produce either a very dilute urine or a very concentrated urine, depending on transport properties at the time and hormones that are surrounding the nephron. Okay? Yeah.
So the peritubular capillaries, those are around the cortical nephron?
Yes.
And then the vasorecta is around the juxtamedullary.
Perfect. Okay? So the peritubular capillaries around the cortical nephron, and the vasorecta surrounds the juxtamedullary nephron. Yes?
So on the juxtamedullary nephron establishes an osmotic gradient. What is the meaning for this?
Yep. So essentially, it still has the capability of modifying the luminal fluid inside. It just doesn't participate in establishing that osmotic gradient. Yep. Okay. All right. So that's just kind of a down and dirty. So you guys understand this figure now. You know the difference between the cortical nephron and the juxtamedullary nephron, and the peritubular capillaries and the vasorecta. Okay, so that's important to understand. Note that this is the collecting duct. I'll talk about the anatomy of the nephron in just a minute, but there's several nephron, that deliver the ultrafiltrate into the same collecting duct. That's why it's called the collecting duct. So this is just cut off for simplicity's sake, but you can see this is the distal tubule of several other nephron that basically deliver their ultrafiltrate into the same collecting duct. So that's important to understand as well. Okay? All right, and understand too the top portion here where the glomeruli are. This is the cortex region, and this is the medulla or the intermedullary region. So knowing how the nephron are kind of situated in the kidney is also really important. Okay, just one more thing about this figure. Up here, the osmolarity is about 300 to 320 milliosmoles up here. Down here, remember with those juxtamedullary nephron, it could be as high as 1,200 to 1,400 milliosmoles. Okay, so I think you guys can understand this now too. I'm going to start kind of putting things, making connections and putting things together. You can have vasopressin on board. You guys know a little bit about vasopressin now. But without the osmotic gradient, I mean, those channels, those water channels can be right inserted into the membrane, ready to go. But without a driving force, water's not going to be able to be reabsorbed. You have to have the driving force, which is the osmotic gradient, to actually move water out of the nephron and back into the body. Okay? So the water channels, they're the pathway, right? That's about conductance, if you guys remember that. That's providing a pathway. But if you don't have a driving force, there's going to be no movement. That's how some of the diuretics work. Okay, so we're going to be talking about some of the diuretics in just a minute too. All right, so just a down, quick and dirty of the anatomy of the nephron here. Okay, so we'll just go through it quickly, but filtration happens at the glomerulus. Okay, and we kind of, went through this pretty quickly, but you do know some of the structures now. This red one is symbolizing the afferent arterioles. The blue, and it shouldn't be blue because it's not deoxygenated, okay? These are the efferent arterioles. And inside, these are the glomerular capillaries, where all the filtration occurs. And it filters out of the glomerular capillaries, number one, because there's very high hydrostatic pressure associated with the glomerular capillaries, forcing, right, the plasma out, the ultrafiltrate out into what's called Bowman's space, So inside here is called Bowman's space. Not you, Kelly. Kelly Bowman. Okay. Okay, so this is Bowman's space, and then it gets filtered into the proximal tubule. So this whole structure right here is called Bowman's capsule. And then the ultrafiltrate is delivered into the proximal tubule. Okay? The proximal tubule is all about reabsorption. The bulk reabsorption happens in the proximal tubule. You want to be able to recover all of your essential amino acids, your nutrients like glucose, most of your electrolytes, 85% of your bicarbonate, 70% of sodium and potassium, and 70% of water is all reabsorbed. Now, this is, I've still been getting questions today about the difference between reabsorption and secretion. I know that's kind of an elusive concept right now, but keep practicing it. I always think about the intestines, the gut. When you absorb calcium from the small intestines, you're pulling calcium out of the small intestines, and you're bringing the calcium into your blood. That's called absorption of calcium. Think about it the same way with the kidney. Reabsorption is the same thing. You're pulling molecules out of the nephron, and you're reabsorbing them back into the body, back into the blood, to recover them so that you don't lose them. You don't want to lose them from your body. Okay? So that's how to think about reabsorption. Secretion is the opposite. Secretion is when you transport molecules from the blood, from the interstitial fluid, and you transport them into the nephron to ultimately, if they're not reabsorbed, to be excreted out of the body. Okay, that's called secretion. Okay? All right, so the bulk reabsorption happens in the proximal tubule. The thin loops, they're highly permeable. So that's one thing to remember. The thin loops are highly permeable. They basically allow for solutes to move in and out of the nephron, in and out of the loop of Henle. So imagine what it would be like to be traveling inside the nephron as you encounter more and more hyperosmotic environments. What's going to happen is water is going to flow out. There's going to be a driving force for the movement of water out of the nephron into that very hyperosmotic environment. Okay? So water's going to flow out. Solutes are going to equilibrate. They're going to move back and forth. And so what's going to happen at the bottom of the loop of Henle, the inside and the outside of the nephron is going to have very high osmolarity. Okay? 1,200 could be 1,200 milliosmoles. All right. So here's where it gets interesting. As you actually flow back up towards the cortex, the thick ascending limb is a very special part of the nephron. This is where all the active transport occurs. You're actively pulling out sodium and chloride out of the nephron. So you have my favorite co-transporter, sodium, potassium, 2-chloride co-transporter, pulling an enormous amount of salt out of the nephron. However, water cannot follow in this particular instance. So the water stays in the nephron, but this is what's essentially establishing that osmotic gradient. You're going to get more salt pulled out of the thick ascending limb. In the very beginning, there's going to be a lot of salt in this area, less and less and less as you move up the thick ascending limb. That's basically establishing that osmotic gradient with all of those juxtamedullary nephrons. You can imagine that's what's responsible, that active transport. Does anybody remember what that is called? Does anybody remember? It starts with countercurrent. What's that? multiplier. This is what's called a countercurrent multiplier because it uses active transport to pull the salt out. Why is it active transport? This is a co-transporter, but it's basically pulling salt out, my favorite co-transporter, across the apical membrane, but it has to get across the basolateral membrane. That's where the pump comes into play, right? The pump is using ATP to remove sodium across the basolateral membrane and into the blood. So that's why this is considered active transport, because ultimately you have to have the pump, which uses ATP. It's a primary active transporter, okay? So you're using active transport to pull salt out, mainly here. Most of the salt is pulled out in the beginning of the thick ascending limb, less at the top. This is essentially called the diluting segment. The thick ascending limb is the diluting segment. Okay, so here at the top, at the distal tubule, this is where all of that intrinsic mechanism, that regulatory mechanism, occurs. We called it autoregulation. Does anybody remember the exact name of that regulation, that intrinsic regulation? It wasn't myogenic. Yeah, no, it has something to do with a tube and the glomerulus. It's called, yes, tubuloglomerular feedback. Tubuloglomerular feedback. So this is where all that tubuloglomerular feedback occurs. Okay, so then going on to the distal tubule, remember the distal tubule and the collecting duct are about fine-tuning. This is about fine-tuning that last urine, that ultrafiltrate. So essentially with the distal tubule, you're still getting, that should probably read sodium chloride reabsorption to be, formal about it. It shouldn't say absorption, it should say reabsorption. In this case, though, potassium and acid protons are being secreted. Bicarbonate is being reabsorbed. And water is being regulated by vasopressin, arginine vasopressin, ATP. It's also called antidiuretic hormone. All right, same with the collecting duct. We're getting sodium chloride reabsorption, potassium proton secretion. This is where most of the bicarbonate is being reabsorbed. And I have to tell you a funny story. Maybe in biology, CBS students may know Sarah. What's her last name? She runs the labs. So there's a number of different labs that are in the NCB building. Sarah Neal. Anyway, when I was first developing the human biology lab, I was doing a lab related to kidney. And I wanted them to eat something that was very, had a lot of bicarbonate in it. And it does work, I do have to say. If you eat a lot of bicarbonate, you will change the urine composition and basically excrete a lot of bicarbonate, which would increase the urine pH. So I wanted the students to eat baking soda. That's so bad. Okay, but none of the students wanted to do that. So Sarah, the lab coordinator, choked down baking soda, and it did change her urine composition. She was quite a trooper, I have to say. Then we realized that you could eat Tums and get the same effect. Okay, so you guys can try it at home if you have one of those sticks, right, that measures pH. If you eat a lot of Tums a couple hours later, your pH of your urine will go way up because you're excreting bicarbonate is what you're doing. Normally, however, you'll completely reabsorb all the bicarbonate in the collecting duct. But if you have an overabundance of bicarbonate, you'll excrete it out. Okay, funny story. All right, and then water and basically urea are also affected by vasopressin in the collecting. Okay, you guys feel pretty good about that? The overall anatomy of the nephron tells you a lot about function as well, but it's a good thing to know. For those verbal learners, if you guys need some text, this is just saying the exact same thing. On the left-hand side, these are all the different parts of the segment, their major functions, and some of the controlling factors, okay? We also talked about the differences between aquatic and desert animals. Remember, the desert animals have a lot of juxtamedullary nephron, and they're actually really long, to the point where they have very thick intermedullary regions and very pronounced renal papilla. Very pronounced renal papilla, as you can see right here, and the renal pelvis. All right, so that's just kind of a comparative look at different organisms and how the morphology of the kidney and how it affects the ability to concentrate urine. Okay, so you guys feel pretty good about this? You feel pretty good about these four processes: filtration, reabsorption, secretion, and excretion. I have to say with the excretion, don't memorize all of those compounds. I just want you to know what excretion is. There's excretion of certain nitrogenous wastes like ammonium ions and urea, but don't memorize all of those other ones, okay? There's not going to be any questions basically on those compounds, okay? Okay, so here is another part of these four processes. I did want to talk about filtration, just briefly. There will be some questions on net filtration pressure. So again, very simple calculation. Make sure you know how to calculate net filtration pressure. Okay. The hydrostatic pressure in the glomerular capillaries is very high compared to other capillaries in the body. It's essentially double. It's like 60 millimeters of mercury. And that's the driving force to make sure fluid completely runs through the nephron and then out through the minor calyces and the major calyx. Okay? So we have 60 millimeters of mercury inside the capillaries, only about 15 millimeters of mercury in the lumen. The oncotic pressure inside the glomerular capillaries is about 30 millimeters of mercury. And there shouldn't be any large blood proteins in Bowman's capsule. Okay? There should not be any blood proteins in Bowman's capsule, so that's going to be 0. So if you calculate this out, then at the bottom of this slide, that's the equation. Net glomerular filtration pressure is equal to the hydrostatic pressure in the glomerular capillaries minus the hydrostatic pressure in Bowman's space minus the oncotic pressure in the glomerular capillaries. But since the oncotic pressure in Bowman's space is 0, that's left off the equation. The thing to remember is essentially that if it's a positive number, filtration is going to occur. If it's a negative number, which hardly ever happens, that would be really bad. That would be absorption, reabsorption. Okay, so did you guys understand these figures here? These figures? Essentially, it's kind of counterintuitive in some ways. I think it's very straightforward when you think about constriction of the afferent and constriction of the efferent. When you're talking about dilation, it gets a little bit more counterintuitive. So just remember, with a dilation of the efferent arterioles, you're going to decrease the pressure in the glomerular capillaries, which is going to decrease glomerular filtration rate. If you dilate the afferent arteriole, it's going to do the opposite. Increase the hydrostatic pressure in the glomerular capillaries and increase glomerular filtration rate. Okay? So that's filtration. I think you guys understand that pretty well. This is a figure specifically giving you information about reabsorption and secretion. I think you guys have a good idea of how to think about that now. You will see a question like this on the exam. It'll be, just like our top hat question, it'll be something like, which one of these figures is filtered at the glomerulus and reabsorbed and excreted? Reabsorbed and secreted. I'm sorry, reabsorbed and excreted. Reabsorbed and excreted. Which one is that? Y. Yes. It is substance Y. Now, just, for your own, remember, PAH acts like substance X. And PAH is a good marker for, you guys remember PAH? Renal plasma flow, right? Because it's filtered at the glomerulus, but what's not filtered still ends up in the urine because it's strongly secreted. So it's a good measure of renal plasma flow. Okay, here's where I know this is a source of huge confusion. Okay, so let's go through these transport properties. I think this is worth your while. If you kind of blow up these slides, it gives you a little text. But I'll tell you right now, the top one is what's happening in the proximal tubule. The middle one is what's happening, that's the transport properties in the thick ascending limb. And the bottom one is the transport in the collecting duct. So this is kind of comparing and contrasting the transport properties in 3 different segments of the nephron. And I'll give you some tips on how to recognize it and how to think about it. So this one, you should be able to recognize it is an proximal tubule because the first thing that's going to tip you off is that sodium glucose cotransporter. Okay, so you know that the bulk, all of the reabsorption, I'm just going to say this, all of the reabsorption of glucose and amino acids, 100%, happens in the proximal tubule. Okay? So when you look at this and you see sodium glucose cotransporter, that should automatically tip you off that this is the proximal tubule. So it uses the sodium gradient, because remember, high sodium outside, low sodium inside. It uses the sodium gradient to move glucose against its concentration gradient. Glucose then moves across the basolateral membrane by this facilitated diffusion transporter. It can just go down its concentration gradient. Okay? Let's go to this equation right here. You're going to see this one over and over and over again. So, yes, this is the first time you've seen this equation here, but you'll see it with the respiratory system in red blood cells. You'll see this in the stomach, in the parietal cells, how acid is transported into the stomach after you eat a meal. So the equation is exactly the same. Carbon dioxide can freely move in and out of the membrane. Okay, so carbon dioxide and water, they combine to form carbonic acid. And the thing to remember is with every carbon dioxide molecule that enters into the cell, 1 proton is secreted and 1 bicarbonate ion is reabsorbed. It makes a lot of sense. Yes. Yes, absolutely. For every carbon dioxide that enters into the cell, you can see it combines with water, is converted to carbonic acid, and then it breaks down into protons and bicarbonate. So for every For every carbon dioxide that enters into the cell, 1 proton is secreted and 1 bicarbonate ion is reabsorbed. It makes a lot of sense because on a daily basis, you produce so much acid. Just by walking around, all the lactic acid you produce from your muscles, just sulfuric acid from just metabolism, cellular metabolism, you have to manage that acid load. So the way to manage it is to secrete the protons back into the nephron to be excreted and to recover your buffer, right? That makes sense, to recover the buffer and transport it back into the blood. That helps regulate blood pH. Okay, so remember all of the sodium, the net transport is moving sodium from the inside of the tubule, the nephron, to the blood in red. And chloride is going to follow through the paracellular pathway. So you're going to get a lot of salts being reabsorbed. Remember, sodium and potassium are all going to be reabsorbed along with chloride. All right? So that's the main thing here. There's a potassium channel up there to basically make sure that the pump keeps working, right? Because there's very low potassium outside. You need to have that channel to make sure that the pump isn't rate limited. You know what I mean? This is basically feeding the pump so it doesn't stop, okay? And then there's some potassium channels that also provide driving forces for the movement of sodium in. Okay? All right, so, but the main thing here is the sodium glucose transporter and this equation here. Protons are being secreted, bicarbonate ions are being reabsorbed. Okay, so what happens with diabetes? What happens with diabetes? You have such high blood glucose levels that the glucose overwhelms the kidney at the proximal tubule. It can't reabsorb all of that glucose. And that glucose ends up in your urine, right? That is one sign of diabetes mellitus. That glucose overwhelms the proximal tubule. You can't reabsorb it all, and some of it ends up in the urine. Okay. Which is abnormal. Shouldn't have glucose in your urine. Okay, let's move on to this figure. This is what's happening in the thick ascending limb. You have on the apical side, like I said, my favorite cotransporter, the sodium-potassium 2-chloride cotransporter. It is bringing an enormous amount of salts into the cell. And all of these salts are moving across the basolateral membrane by different cotransporters. You've got the potassium chloride cotransporter, chloride channels, and the pump that's moving sodium across the basolateral membrane. So all of those salts are moving from the tubular lumen into the blood supply. There's a net movement of chloride across the transcellular pathway, so sodium is actually going to follow along the paracellular pathway. But water is not allowed to follow all of that solute movement. That's why this is again called the diluting segment. So the tip here is to see that this is the sodium-potassium 2-chloride cotransporter. Automatically that should tip you off that this is the thick ascending limb. Okay, the thick ascending limb. And this is active transport, again because of the pump here, is using ATP to basically move all of that salt across the epithelial, the monolayer here. Now remember, this is for simplicity's sake. You still have carbon dioxide and water is being converted to carbonic acid and then broken down into protons and bicarbonate. For every carbon dioxide, 1 proton is being secreted and 1 bicarbonate is being reabsorbed. So that takes place here as well in the thick ascending limb. But again, just the take-home message is this cotransporter. This should tip you off that this is the thick ascending limb. Now remember the drug Lasix, right? Furosemide. This is what they give horses to control nosebleeds and edema, okay? This is what they give people when they have heart failure or super high blood pressure, right? It's a powerful diuretic. Powerful. It's probably the most powerful diuretic out there. But you shouldn't be on it for too long because it's going to disrupt the osmotic gradient, number one. It's going to disrupt the osmotic gradient. You're going to lose an enormous amount of electrolytes, sodium, potassium, and chloride. You're going to lose to your environment. You're going to pee that away. Okay, so that's not good. But I want you guys to kind of think about this as to why it's a diuretic. Okay, why is this a diuretic? Let me just show you something. I didn't have a chance to kind of go through this in detail in class, but I think this is an important concept here. So I'm going to just put this on mute. This is going to give you a really good idea of how diuretics work. So on the exam, when you get this application type problem and you're given, say, a diuretic, this is going to give you a lot more insight as to how diuretics actually work. Okay? So we have the glomerulus. Okay? Alright. And the fifth ascending limb. Oops, I'm going to put the distal tubule over there. Alright. And then the collecting duct. This is a horrible picture. Collecting duct. All right. This is what we're talking about right here. All right? Okay. So normally, when that sodium-potassium 2-chloride cotransporter is working properly, it's pulling an enormous amount of salts out in the beginning. Okay? And then as you move up, less and less and less and less. Okay? So you can see that this is establishing that osmotic gradient. So if you block it with, say, Lasix, furosemide, again, one of the most powerful diuretics, you're blocking this process. Okay? And all of the salt is actually now staying inside the nephron. Okay? And what happens? It actually gets delivered into the collecting duct. Right? All of the salt gets delivered into the collecting duct. Okay? How is that going to affect the osmotic driving force? Now let's say vasopressin is on board, and you can reabsorb water, but instead of just a few solutes inside the nephron, now you have all of the salt inside the nephron. It's going to disrupt that osmotic driving force. And you're not going to be able, even though you have water channels in the collecting duct, you have a pathway for water, you've lost your driving force for the ability to reabsorb water. So all the water stays inside the nephron. You can't concentrate. You can't pull that water out. And you lose everything. You lose all of that solute. You lose all of that water to your environment. It gets excreted out. That's why it's such a powerful diuretic. You've disrupted the osmotic driving force to pull water out. And so all the water, you lose all that water, but you lose all those electrolytes too. Yeah?
So why would you do this with someone with heart condition?
Yeah, usually with heart failure, it's a problem really with the pumping action. The heart doesn't pump as well as it used to. So, in effect, what happens is you start getting edema in your extremities. Usually people that have heart failure, their ankles are huge, right? Yeah, the swelling is really, really bad. So just to get rid of a lot of the excess fluid in your body, it's a good Did you have a question? You look like you're...
We were trying to figure out the best way to describe this.
Okay, secretion is basically where you take molecules from the blood and you deliver it into the nephron. This is secretion. Excretion is this arrow here. When you've actually eliminated urine from your entire body, it leads through the urethra. Really, it's a fancy name for peeing. Excretion, micturition. It's basically fancy names for urination. Okay. All right, so let's continue on. I think you guys have a really good idea of how diuretics work now. I think so. Okay. All right. So the next one here is the collecting duct. And it's very simple, but I think because you guys know aldosterone now, if you just saw the simple figure, I would think immediately the collecting duct and aldosterone. Because aldosterone is the steroid hormone that upregulates these epithelial sodium channels. And the more sodium channels that are inserted in the membrane, the more sodium enters into the cell. It increases the turnover rate of the pump, and then more potassium is secreted. Okay. Yep. So aldosterone is the hormone, the steroid hormone, that basically upregulates these epithelial sodium channels. They insert more and more sodium channels in the apical membrane of these cells. That increases the amount of sodium that enters into the cell, increases the turnover rate of the pump. It basically increases the amount of potassium that is transported into the cell and the amount of potassium that is basically secreted. Potassium is then transported from the cell into the nephron. Okay? So, a diuretic, just like we talked about here, kind of see it down here, right? If you block that sodium channel with a molecule called amiloride, the same thing happens. All of that salt, the sodium, stays inside the nephron, and it disrupts the osmotic driving force. So you get sodium that's excreted and water that's excreted. But in this case, not a whole lot of potassium. That's why it's called a potassium-sparing diuretic. It basically inhibits this sodium channel right here. So all the sodium stays in the nephron, all the water stays in the nephron, but you don't get a lot of potassium secretion as a result. So it's a potassium-sparing diuretic. Yeah? Not always. That's a good question. Sometimes it's secreted and then reabsorbed. Yeah, that's kind of tricky. So I would say a lot of times it is, right? But it's not. That's not an absolute statement. Yep. Yeah, I would say in this case potassium is excreted because it's in the collecting duct. It's in. Yeah. Okay. So do not memorize this. Do not memorize this. Do not memorize this. But do know this. Okay? So this is what I will ask you about. I'm not a biochemist. I know creatine and creatinine, but that's about it. So, but this is the physiology of excretion. This is what happens with micturition, which is, again, a fancy name for urination. Do know the different muscle types. One is regulated by the parasympathetic. Those are the detrusor muscles that surround the bladder. One are actually here at the internal urethral sphincter that's regulated by the sympathetic nervous system. But nothing should happen until you relax the external urethral sphincters. That's all under voluntary control, under skeletal muscle control, okay? Okay, so it's important to understand that during filling, those detrusor muscles are relaxed, okay? So the parasympathetic neurons are inhibited. Then during micturition, during peeing, those are actually stimulated. The detrusor muscles contract, which helps you eliminate the urine from your body. Sympathetic causes, basically it's stimulated during filling and then relaxed during micturition. Same with the skeletal muscle. It's stimulated during filling, and then when you go to the bathroom, you physically inhibit those and relax them so that you can urinate. Okay? All right. All right. It's kind of good to see and maybe get a little bit more information, put some of those pieces of the puzzle together. Let's go to lecture 12. Yeah, we're doing pretty good on time. Any burning questions? You feel pretty good about that, you guys? Okay, 12. Okay. Okay, so if I was to erase this, give you a roadmap of where we should go from here, the main ideas are intrinsic mechanism, intrinsic regulation. That included myogenic regulation, tubulo-glomerular feedback, and we talked about meningeal cells. Then I took a break, a minute, and I talked about the difference between countercurrent multipliers and countercurrent exchangers. That kind of set me up to talk about the osmotic gradient and extrinsic regulation, because you need to know that before you get into extrinsic mechanisms, extrinsic regulation. And we concentrated on mainly hormones. Okay, we did talk a little bit about the sympathetic nervous system, but mainly we talked about vasopressin, acronym for that is arginine vasopressin, or AVP. It's also called antidiuretic hormone. Or ADH, antidiuretic hormone. Then we talked about aldosterone. And this one always sends students into quite a crazy frenzy. Renin-Angiotensin pathway. But it's good to know. In fact, I had a CBF student come up to me after that lecture and show me the MCAT exam that had that particular pathway on it. So, just so you know, that is something that should be studied for you guys. Renin-Angiotensin. Now remember, the other part of this is the renin-angiotensin aldosterone arm. So angiotensin 2 actually promotes the secretion of aldosterone from the adrenal gland. Okay? And then finally, atrial natriuretic peptide. Natriuretic. Natriuretic peptide. Okay? And that really is the end of lecture. This is just a quick roadmap of the entire lecture 12. It's a lot to it, okay? But if you can master this, then you should do fine on the exam. Okay, so this was just review. Here's where we get to myogenic versus tubuloglomerular feedback. There's a lot to tubuloglomerular feedback. In fact, there's a paper on Moodle that talks, I think there is, it says not going to be on your exam. But if you really want to know more about tubuloglomerular feedback, it's a pretty extensive feedback mechanism, okay? But for our purposes, when we're just learning about this, I don't want to overwhelm you. This is the two charts to just understand. And if you want to put some arrows, I had some students come up after class and they wanted to know how to read these two graphs. We'll go through it again so that it's clear to you. Okay, so let's just take myogenic regulation first. It's actually fairly simple, even though reading this flow chart can be maddening in some ways. So I would put a little like a 1 with a circle around it and put an arrow that goes down in this direction. Okay? A 1, just a 1 symbol, and a big arrow that goes in this direction. And then I would put a 2 right here and then go in this direction. Go to your right and then down. So we're going to read the first one and then the second one. All right, so the way to read this is in the face of an increase, like a surge of high blood pressure, where the mean arterial pressure goes up. What's going to happen? This is going to be the result, is you're going to get an increase in the afferent arteriolar pressure, an increase in the glomerular capillary pressure, an increase in glomerular filtration pressure, right? That's really the hydrostatic pressure in the glomerular capillaries, and ultimately an increase in glomerular filtration rate. Okay, that's just the consequence of an increase in mean arterial pressure, okay? But the compensatory mechanism, that reaction to the high blood pressure, is number 2. So now we're going to read it this way. We're going to go to the right and then down. That's the compensatory mechanism. That's going to compensate for the surge in high blood pressure. So what happens immediately is there is a stretch with the high blood pressure. There's going to be a stretch of the arteriolar smooth muscle. That's going to increase constriction. So if you really want to understand the mechanism responsible for that, think about what happens when you stretch smooth muscle. You're probably going to stretch those cytoskeletal elements. There's going to be stretch receptors that are stimulated. You're probably going to get an influx of calcium into those smooth muscle cells. Remember, with smooth muscle, if you get an increase in calcium, you're going to get constriction of those smooth muscle cells, right? We learned about that with the muscle section. Increase in calcium causes constriction. Okay, so with a stretch of the arteriolar smooth muscle, you're going to get constriction of those smooth muscle cells, constriction of the afferent arteriolar vessels. The resistance is going to go up because the radius is going to decrease when you get constriction. You're going to get decrease in glomerular capillary pressure, decrease in glomerular filtration pressure. That's going to decrease glomerular filtration rate to keep glomerular filtration rate in homeostatic ranges. All right? So the first one Going down is just telling you something about what happens in the face of high blood pressure, a surge of high blood pressure. Number 2 is the compensatory mechanism. That this is the myogenic regulation, the autoregulation as a result of the increase in mean arterial pressure. Do you guys get that? Okay, pretty. I see a lot of heads going up and down. So, anybody confused? Are you guys confused? No. Okay, good. Yeah, you started going good. Okay. All right. So that's myogenic regulation. Okay, so let's go with tubuloglomerular feedback. The way to read this chart is to go. This one's a little bit more, I think, easier to read. You go straight down, and then to the right, and then up towards. So you're going in a counterclockwise direction. Okay? So, again, in the face of high blood, yes?
So the left side, increasing the pressure, that was.
What's that?
So if the mean arterial pressure increases,
Yep.
increase in blood pressure.
Yeah.
So, I guess I was trying to confuse blood pressure. Side stretching.
Okay. So this would be a surge in high blood pressure. Essentially, what would happen, and I'm talking with a surge in high blood pressure, maybe with exercise, when you increase heart rate and stroke volume, you're going to get an increase in cardiac output and mean arterial pressure. So with a surge of high blood pressure, there's going to be a very acute stretch, right, of those afferent arterioles. And that's going to stretch the smooth muscle, activating those stretch receptors, probably I'm sure it involves calcium. You're going to get a compensatory constriction of the smooth muscle cells that surround the afferent arterioles. Okay, that's going to help with the autoregulation to make sure that the glomerular filtration rate stays constant. Does that make sense? Yeah, okay. So with tubuloglomerular feedback, what you have here is an increase in mean arterial pressure, an increase in the afferent arteriolar pressure, increase in glomerular capillary pressure, an increase in the glomerular filtration pressure, and ultimately an increase in glomerular filtration rate. Okay, so here's where I think that there's some confusion, like what happens, what happens in between the increase in glomerular filtration rate and here the increase in flow at the macula-densa cells. Okay, so the more ultrafiltrate, basically that's filtered at the glomerulus, the more flow that's going to happen in the distal tubule. You're going to get an increase in the flow that's going to zip by the macula-densa cells. Okay? So an increase in glomerular filtration rate is going to translate to an increase in flow in the distal tubule. Okay? Now, there is a huge gap, but for our purposes, this is all I want you guys to know. There's chemical signals. We don't know what that is. It's not defined. It's some kind of paracrine signaling. Para meaning next to. These are some kind of chemical signals, paracrine signals, that are released that cause constriction of the afferent arterioles, increase in resistance of the afferent arterioles, and a decrease in glomerular capillary pressure. Okay? Okay, so I'm going to fill in some of the gaps. I talked about this briefly in class, but essentially it is those sodium, potassium, 2-chloride cotransporters that are detecting the flow. If the flow is too great and it's zipping along past those macula-densa cells, there's not enough dwell time for the salt to bind to the sodium, potassium, 2-chloride cotransporter. So that's going to release those chemical signals. It also happens when the flow is too slow. You're still not going to get a whole lot of salt that are going to bind to those sodium, potassium, 2-chloride cotransporters. Either way, if the flow is too slow or too fast, you're going to get these chemical signals that are going to constrict the afferent arterioles. Some of the missing pieces here are, you have, let's see, at a loss, right? Adenosine. Adenosine is the molecule. That's part of the chemical signal. Adenosine. But we're not going to go through that whole pathway. That's just way too much information. So for our purposes, just know that it has something to do with the sodium-potassium 2-chloride cotransporter, chemical signals like adenosine, and constriction of the afferent arterioles. Okay? Yes, one to the back.
Can these regulations occur at the same time?
Are these? Yes, they are at the same time, right? All right. The other one we talked about was the meningeal cells, glomerular meningeal cells. This is another intrinsic pathway, autoregulation. So let's just take a look at this particular figure again. Here's where the meningeal cells, there are these hot pink cells in the middle here, right next to the macula densa cells, right next to the juxtaglomerular cells. These are what's called the extraglomerular meningeal cells. Okay, so they're outside the glomerulus, but they're also meningeal cells inside the glomerulus, intraglomerular meningeal cells, that control the filtration slits. Okay, so there's chemical signals by the extraglomerular meningeal cells and intraglomerular meningeal cells. But the only thing that you guys have to know is these meningeal cells are responsible for decreasing the glomerular filtration rate in the face of very high blood pressure, a surge of high blood pressure. And how it does this is it actually creates these glomerular capillary loops, these meningeal loops. So it cuts off the blood supply to a particular part of the glomerular capillaries, reducing the surface area and ultimately filtration. It also has effects on the filtration slits themselves, making those filtration slits smaller, reducing glomerular filtration rate. All right, you guys. Here's the take-home message, why this is so important. You can see here in the face of varying mean arterial pressure, I mean literally from 80 millimeters of mercury to 180 millimeters of mercury, these autoregulatory mechanisms are keeping glomerular filtration rate pretty steady. That is an amazing graph right here. That's because of all these auto-regulatory mechanisms in place. And you can see there's a lot of redundancy. The other thing to take notice, I mean, I think you guys have a good grasp on how to think about the afferent constriction of the afferent and efferent arterioles. But this is a really important graph that lets you know why glomerular filtration rate is important. As glomerular filtration rate decreases, and this is what happens in chronic kidney disease and basically kidney failure. Kidney failure is technically 15 mils per minute. That's a GFR of 15 mils per minute is technically kidney failure. And you can see when you get so low with the glomerular filtration rate, you can't filter these molecules out of your blood anymore. The plasma creatinine levels are very abnormally high. So you're not filtering these molecules and ultimately excreting them. Okay? All right, so then we took a break from this and talked about countercurrent multipliers, how the loop of Henle is responsible for establishing that osmotic gradient, and the vasa recta. Okay, why the vasa recta is organized this way to help maintain the osmotic gradient. I have had students in the past, after the third exam, come up to me and say, I didn't realize that this was the circulatory system, that this was the blood supply, okay? So I know that there's some confusion with these two graphs. This is the nephron, and it is establishing that osmotic gradient. And here's some numbers. You can actually see the osmotic gradient that's established in the inner medullary region, and that's established by those juxtamedullary nephron. This is a graph showing you the vasa recta. This is the blood supply surrounding the juxtamedullary nephron. And it's organized in a U shape like this. If it wasn't, if it was organized in this linear fashion, it would be constantly, right, solutes would be equilibrating into these tubes, and then the blood supply would be just washing it away. It would be constantly eroding that osmotic gradient that was established by the loop of Henle. Instead, it's actually organized in a U shape like this. Now, does anybody remember the name of this one? What's that? It is the vasa recta, yep. Countercurrent exchanger, yes. Countercurrent exchanger. Okay, so the multiplier is the loop of Henle. The countercurrent multiplier is the loop of Henle, because that is the one that is establishing that osmotic gradient by active transport mechanisms. This is called the countercurrent exchanger, because it's all about just passive transport. Okay? Just passive transport. So a long time ago when they taught physiology, they used to use the same term, countercurrent exchanger, for both the loop of Henle and the vasa recta. This is a little bit more precise. It's giving you an idea of which one is using active transport mechanisms and which one is just using passive transport. They're different mechanisms, right? And I always think about the vasa recta when I think about this figure. Think about a red blood cell. A red blood cell that has to travel from 300 milliosmoles to 1200 milliosmoles and back. That is some amazing cell volume regulation. Okay, I think that's impressive. Okay, so the other one that I have to make sure you guys remember is urea. Urea is a very important osmolyte. I can guarantee there'll be a couple questions on urea on the exam. Just a heads up on that. Urea is also regulated by vasopressin. So I think about it this way. Imagine the collecting duct when vasopressin is on board. Vasopressin is going to promote water to be reabsorbed. So a lot of water is going to be moving from the collecting duct out into the interstitial fluid. If urea didn't follow, it might disrupt that osmotic gradient. So urea is an important osmolyte, again, maintaining the osmotic gradient in the intermedullary region. That's the way I think about urea. Okay? And it's one of the reasons why vasopressin also regulates urea. Vasopressin regulates water reabsorption and urea. Okay, so now we're at the last four. This shouldn't take us more than maybe 15, 20 minutes just to go through the hormonal regulation. But this is the very last part of it. So if you know how vasopressin works, how aldosterone works, you will probably have to memorize the Renin-Angiotensin System, just like you did the G protein coupled regulation. But once you understand it, it's not too bad. And then atrial natriuretic peptide, which is the only diuretic. Vasopressin, aldosterone, and the Renin-Angiotensin System, they're all antidiuretics. They all basically are mechanisms to promote water conservation, basically water reabsorption. So it helps you concentrate urine. Okay, so you guys did your severe dehydration, right? Case study, which was an important part of this. Let's start here. With severe dehydration, your blood osmolarity is going to be abnormally high. Okay? You're going to have very little body fluid, but you're going to have very high osmolarity in the blood. Okay? So, essentially, what happens is that high osmolarity, that high blood osmolarity is detected in the hypothalamus by the organum baculosum of the lamina terminalis, the subcortical organ, and the median preoptic nucleus. You don't need to know that. It's good information, but that's not going to be on your exam. Just know. I just want you to guys to get the bigger picture. There's osmoreceptors in the hypothalamus. That's what you need to know here. Osmoreceptors in the hypothalamus. Okay? What that does when these osmoreceptors are triggered is it promotes a very profound thirst behavior. And it releases vasopressin from the posterior pituitary gland. And you'll learn a lot more about the posterior and anterior pituitary gland in a few weeks after the third exam. Okay? So now vasopressin is released into the circulatory system. It travels all the way to the kidney, to the collecting duct. Notice that it's on the basolateral membrane, right? This is where the blood supply is. Vasopressin is a peptide hormone. So the receptor is on the plasma membrane. That means that the peptide hormones are basically hydrophilic. They bind to the receptor. This receptor is a GS-coupled protein. I always have to laugh about this too, you guys. When I was defending my master's, right, I had an oral defense, and they asked me about vasopressin. And they said, which G protein is coupled? I go, GQ? They go, no. And I go, GI? They go, no. I go, oh, okay, GS. It was my last guess. It was really super embarrassing. So I never forgot that vasopressin receptors are GS coupled in the kidney. Super embarrassing. It happens to us all, you guys. It's all a learning process. All right, so vasopressin binds to its receptor, GS-coupled cyclic AMP increases in the cell. It activates pKa. It basically is a very quick response. It happens in literally seconds. It promotes the insertion of aquaporin 2 channels on the apical membrane, right? Aquaporin 3 and 4 are always on the basolateral membrane, but once, this is the inducible response, once aquaporin 2 is inserted into the apical membrane, now you have a pathway for the movement of water. Okay? So this allows you to concentrate urine. Now the water can follow the solute movement. There is an osmotic driving force for the movement of water out of the nephron into the interstitial fluid. So remember, as the fluid flows through the collecting duct, as it flows down into more hyperosmotic, right, more hyperosmotic environments, you're going to keep increasing that osmotic driving force for the movement of water out if there's a pathway for water. Okay? The diuretics, they disrupt that osmotic driving force because it delivers a lot of salts inside the nephron, and it disrupts the osmotic driving force for the removal of water out of the nephron, or the reabsorption of water. Okay? Does everybody kind of get that, how diuretics work? It's really all about osmosis, right? Diffusion and osmosis, which is why I start off with that in class, our very first sections. Okay? Kidney's all about diffusion and osmosis. All right, so again, for more verbal learners, this is the same thing. It just has some text associated with it. Again, this is just hitting home how to think about it. The blue on the left-hand side is what happens when vasopressin is on board. You have a pathway for water, and you can see as the fluid in the collecting duct moves into more hyperosmotic environments, more and more water is pulled out of the nephron and reabsorbed into the body. If vasopressin is not on board, if it's not around, in the case of diabetes insipidus, right, you have a head injury, your hypothalamus can't produce vasopressin, this is what's going to happen. All that water is going to stay inside the nephron and be excreted. Okay? You're not going to be able to produce a concentrated urine. It's not going to be possible. And take a look at this. I think this is pretty profound too. You can concentrate urine. Urine can be as concentrated as 1,200 milliosmoles. Without vasopressin, only 65 milliosmoles. That's a huge range in basically the specific gravity of urine. Urea. This always seems like a trick question. Urea is regulated by vasopressin as well. Okay, remember urea is an important osmolyte that helps maintain the osmotic gradient. Okay, next one, aldosterone. Anybody remember what type of hormone this is? Aldosterone. Steroid hormone, yes. This is a steroid hormone. The response is awfully slow. Because the receptor is inside the nucleus, aldosterone freely moves across the plasma membrane, freely moves into the nucleus, and promotes transcription. So you get transcription and translation of these epithelial sodium channels. And it takes about 24 hours. It is not a quick response. It's actually a very slow response. Once you've seen this one already, once you increase the number of those epithelial sodium channels, you're able to transport more sodium into the cell, which increases the driving force, or not the driving force, it increases the turnover rate of the pump. So you get more potassium that's transported inside the cell, and more potassium is secreted. Okay. Any kind of inhibitor of the epithelial sodium channel is going to be a potassium-sparing diuretic. Now remember, water is actually going to follow that sodium movement, but only if vasopressin is on board. Okay, so water will follow that sodium movement, but only if vasopressin is on board. Okay, high potassium levels inside the blood also promotes aldosterone secretion from the adrenal gland. I was going to ask you that question too. Aldosterone is basically secreted from the adrenal cortex region. So this is a nice flow chart. An increase in potassium intake increases your blood potassium levels. That actually initiates aldosterone secretion from the adrenal cortex. You get high plasma aldosterone levels. That promotes potassium secretion, right? And you get increase in potassium excretion. Remember, potassium is one of those electrolytes that should be tightly regulated. It can cause arrhythmias and death if potassium levels get too high in the blood. Okay, you guys good with that? This is the next one, the renin-angiotensin, aldosterone arm. I like this one because it really, if you guys have kicked around the term. A lot of people know ACE inhibitors, but they don't exactly know what they are, right? This tells you exactly what ACE inhibitors are. I would, at this point, you might talk to your parents or your grandparents. They're probably taking high blood pressure medication. I ask people all the time because I'm not shy. They tell me what diuretics they're on. They're usually on diuretics. They might be on ACE inhibitors. They might be on beta blockers. I mean, they'll go ahead and tell you. And then you know exactly what all of that means now, which is cool. Okay, so the renin-angiotensin system. Know that angiotensinogen is produced by the liver. Angiotensinogen is the precursor molecule. Now this figure says renin is excreted from the kidney. But where is it secreted from? Does anybody remember this from your severe dehydration exercise? Where's renin coming from in the kidney? There are cells that surround the afferent arteriole. What are they called? I hear it. Juxtaglomerular cells. The juxtaglomerular cells are secreting renin. I'll show you where those are. Here they are. Juxtaglomerular cells in, what is that, cyan blue? Juxtaglomerular cells. They're not that color. Okay. All right. So the renin is actually being secreted from the kidney from those juxtaglomerular cells. Renin is an enzyme that converts angiotensinogen to angiotensin 1. But that's not the active form. Now, this symbol, a lot of people don't realize this, but this cylinder is symbolizing a blood vessel, And it's in these endothelial cells that line the blood vessel where you're going to find ACE. That's the angiotensin converting enzyme, ACE, angiotensin converting enzyme. It is in the blood vessels, in those endothelial cells. That's where the enzyme is. So as angiotensin 1 flows through the circulatory system, it encounters those enzymes that convert it from angiotensin 1 to angiotensin 2. And that's the active form, angiotensin 2. And it does a couple of different things. Like the name suggests, angio refers to the blood of vessels. Tensin is going to refer to the constriction. It is a powerful vasoconstrictor. Okay? Powerful vasoconstrictor. What that's going to do is it's going to increase total peripheral resistance. And remember in Ohm's law, that's going to increase mean arterial pressure. So this happens in the face of low blood pressure, like severe dehydration. And the end result is a decrease in blood pressure. Yes. Yes. So from the blood vessel. Okay. ACE is angiotensin converting enzyme, and that is within the endothelium. That's the cells that line the blood vessels. As the angiotensin 1 flows through the blood vessels, it encounters the enzyme ACE that converts it from angiotensin 1 to angiotensin 2. And angiotensin 2 is the active form of the molecule. Okay? So you can see angiotensin 2 does two things. It's a very powerful vasoconstrictor. Okay, so when you constrict these vessels, you ultimately increase total peripheral resistance. Okay, total peripheral resistance. That, you remember, mean arterial pressure equals cardiac output times total peripheral resistance. So if you increase total peripheral resistance, you're going to increase mean arterial pressure. Okay? So you get an increase in blood pressure as a result. Also, it promotes secretion of aldosterone from the adrenal cortex region. And remember what aldosterone does. Aldosterone promotes, here it is, it promotes salt reabsorption, specifically sodium reabsorption. And water does follow if vasopressin is on board. So you get salt, I'm talking specifically sodium. Sodium reabsorption, water reabsorption. In this figure it says salt retention and water retention. Okay? So aldosterone is going to increase, it says salt and water retention, increase blood volume, and that's going to increase blood pressure. Okay? You guys feel pretty good about that? And Renin-Angiotensin Aldosterone. That's on a lot of standardized exams. I'm talking the DAT, the PCATs, MCATs, graduate GRE. I mean, if you're going into the health profession, that's just something that you guys should know. Okay, I would not be doing you guys any justice. I wouldn't be doing this class justice if I didn't teach the renin-angiotensin aldosterone pathway. Okay, finally, atrial natriuretic peptide. You can see here, whenever you get a surge in plasma volume or a surge in blood pressure, whenever you have that happen, an increase in venous return to the heart, and you stretch the atria, that's going to release atrial natriuretic peptide. So it's detecting a surge in high blood pressure, essentially. You get an increase in plasma atrial natriuretic peptide. It basically decreases aldosterone levels. It has effects on vasopressin, so it decreases vasopressin. It decreases the renin-angiotensin system. Has effects on afferent dilation and efferent constriction. Increases glomerular filtration rate, and essentially it promotes sodium excretion and water excretion. It is a diuretic. So I'll say that again. Atrial natriuretic peptide inhibits all of the previous hormones that we talked about, vasopressin, aldosterone, and renin. It also has effects on the afferent and efferent arterioles so that you increase glomerular filtration rate. And ultimately, you're going to get an increase in sodium excretion and an increase in water excretion. It's a powerful diuretic. I told you guys the story about scuba diving, right? Go to depth, you're in a horizontal position, you get an increase in venous return. It never fails, you always have to pee when you get down to about 60 feet or so. It's true. Okay? Good physiology, though. So I'm like, damn, I have to pee. But damn, that's good physiology. All right, you guys, that's pretty much it. I mean, you have to be spent. That's almost 2 hours of the kidney. So I'm glad you guys are here today. I have to say, can you imagine trying to cram for all of this, the renal and the respiratory, before that third exam? Okay. We'll have another review next week. I'll tell you when it'll be tomorrow. Organize that and give you some information about that tomorrow in class. All right, you guys. Any last minute questions? I'll be around. Do that.
Hopefully I don't die.
Yeah. No, don't die. Yes. Don't eat fugu.
Lately, has anyone died from it?
Yeah. Lately, I don't know. But I know people die from it. That's true.